While we're waiting for some other people to show up, let me just tell you a little bit about my own background. I was pretty much in the same situation I think most of you are back when I was in graduate school, because my field was not at all biology. I was a mathematician by profession, and I taught here at the York campus for about 31 years. So mushrooming for me has always been an avocation, and I really got into it when I was going off to college. I grew up in Kansas, but I went to college in Massachusetts, and so it's a long way from Kansas to Massachusetts, and that gave my folks and I a good excuse to take some long road trips and see some of the country. And on one of those, we went through the Great Smokies, and I was really amazed at the variety of mushrooms, all the colorful things, and I thought, boy, these are really neat. So I purchased a little book on mushrooms of the Smokies, and ended up being totally frustrated because nothing that I found was in the book. And it was just more or less a picture book. It didn't have keys. So that kind of stuck in my mind that, you know, there must be some systematic way to figure out what the heck these mushrooms are. And there must be some book out there that actually has, you know, most of them. Hi, come on in. Well, I didn't have time to really look into it when I was in college or even in grad school, but at the end of my years in grad school, I got my dissertation turned in early. I had some time left, and I was at the University of Michigan, which turns out to be a big, or was at least, I think it still is, a big center for mycology. And they had an arboretum there and a botanical garden, and they had little short courses, about 6 weeks, one night a week. They had bird watching, they had wildflowers, and they had one on mushrooms. So I signed up for the one on mushrooms, and that's when I really got hooked. And fortunately, by that point, things were getting a little better in terms of what field guides were available. Because really useful field guides, ones that have keys and that allow you to systematically figure out what something is, have only been available for about the last 20 or 30 years. Before that, they were mostly just picture books, and it was really kind of an unfortunate situation. So anyway, that's basically how I got into mushrooming, and again, I didn't have a lot of time to do it while I was actively teaching, but once I finally climbed the academic ladder, I had a bit more time, and now the last 5 years I've been retired, and so this avocation has become increasingly passionate since I've been retired. Anyway, I'm assuming that most of you have been interested in mushrooms for a while, probably have collected some mushrooms, and maybe have learned from parents, grandparents, or whatever, certain edible species, and you know where to go to find those. And as long as you don't get out of that particular neighborhood or try anything other than those species, you're pretty safe. But if you want to expand your knowledge, then you need to know how to go about it in a way that's safe and systematic, and that's what I want to talk about today. So I'm going to talk about, first of all, what you need to go out and collect mushrooms. So obviously you need a container, and the traditional one is a basket. You don't need one this big. This is a little optimistic for around here, but you know, in good season you can. They're a little heavy. If you want to lighten up things, you can get one of these little fabric things, and then you just pop it open, and you got plenty of space. So I kind of recommend those if you don't want to weigh yourself down. You can certainly get enough in there, whether they're edible or whether they're just ones you're interested in studying. You can certainly get enough in one of these, and then you can just fold it up when you're done. In terms of clothing, one thing we do have to worry about around here are ticks. And so I don't usually wear shorts when I go out here, also because of the brambles. I usually wear a pair of these convertible pants. And then if it is really hot and I need to, I can just take the leg part off. But I start out with these tucked into the socks because of the ticks, because they really are a problem in a lot of places around here. And with the Lyme disease threat, it's something you have to take seriously. If you want a good luck hat, you can always get something like this, you know, that you can refer to when you run onto one of these things. But anyway, in terms of the serious equipment, some sort of staff, it can be just a bamboo pole or whatever. Some sort of walking staff is useful, especially if you're going downgrade. It gives you an extra means of support. But it's also useful for just moving around, getting the leaves out of the way, the plants out of the way. We don't seem to have much problem here with snakes or anything like that, but you could conceivably also avoid contact with something like that. But mostly it's just useful for extra support and for clearing things out. What else? It's very handy to have a little hand lens, and you don't need to spend a lot of money. I got this at a coin shop here in town, and it's perfectly adequate. But there are a lot of features that you can see using a hand lens. This is about a 16 power. You don't want much more than that. You can actually get one that's a little too strong for most uses, but somewhere between 10 and 20 is about right. So these don't cost much, and main thing is, whatever you take with you, you need some kind of lanyard so that you don't lose it in the midst of looking at things. And the same is true for pocket knives. You're going to need some sort of knife. I carry one that has a knife on one side and a saw, little small saw on the other. Don't use that much except with these tough things that grow on the sides of trees, and some of them can be almost impossible to remove without something like that. So I carry that along. Obviously you're going to need some bug spray, and unfortunately we have a lot of poison ivy around here. I don't know how many of you are sensitive to it, but if you are, you need something like this Technu. Most of you know that product. It's an over-the-counter thing. You can find it in any drugstore. But it's really good, either before or after. So if you have a problem with poison ivy, take some of this along, because if you start to itch or whatever, you can put this on and it'll really knock it very quickly. Hi, let me give you a handout here. Anybody else need a handout? Let's see. Okay, 2 more neat handouts here. Okay, I'm going to just pass this back. All right. As to other things, for real small specimens, I carry some film cans, but you could also use something like a pill container. So I use these little things just to keep track of really tiny things that get lost in our usual little bags. So for most mushrooms, what is best for collecting them is a wax paper sandwich bag. Unfortunately, these used to be readily available in grocery stores. They are increasingly hard to find these days. If you know of a grocery that has these things, let me know, because lots of people would really be interested. You can get them online. There is an outfit called mybrands.com that sells these. So you can still get them, but it's a fair amount of work. The advantage, of course, is that they're transparent, and so you can put your specimens in there and you can still see what you have. You don't want to use plastic bags. That is the worst thing you can do, because if you use plastic bags, mushrooms being mostly water will just be one slimy mess by the time you get home. Whereas this, in spite of the fact it's transparent, does breathe, so it will not cause stuff to condense inside. Now you can use paper bags. Paper sandwich bags are small paper bags, but of course you can't see through them. So you're constantly rooting around trying to find maybe one particular specimen. So if you can find wax paper sandwich bags, they're really the thing of choice to put your mushrooms in. It's also useful to have some slips, and on our club forays, at our fancier forays, we sometimes use these. A lot of times on our weekend forays we don't, but it's actually a good idea to have some of these little field slips on which you can note things that are ephemeral, things that you're not going to remember when you get back home or whatever, such as, the habitat where you found it, what the thing was growing on. And if you make these out properly, you can do it so you can just quickly check things off and you don't have to do very much writing. And then there's space left at the top to write down what the mushroom turned out to be if you're successful at identifying it. So those are handy. I usually take a little camera and a tripod, and if you don't know about these handy little tripods, these little so-called jobies, they're very inexpensive, and they not only will act as a tripod, but you can wrap them around things like a fence, rail, or whatever, and you can get them absolutely flat because mushrooms tend to be close to the ground, so you can get these things completely flat. They're really terrific if you're a photographer and you're interested in photographing mushrooms. And then one other thing that may seem sort of silly, but auto supply stores sell these gadgets for working on inaccessible parts of engines. And they're really handy. It's a mirror that telescopes. And so if you don't particularly want to bend over, if you got a little arthritis in the knees or whatever, and you're not sure you want to pick this thing anyway, well, you just hold this down. You can see underneath the mushroom, you can see the bottom side as well as the top side. And you'd be surprised, it's actually pretty handy. And then it just collapses down. It's got a little clip for your pocket, and you can carry it with you. So, as I say, you don't really need to carry all that stuff. That's up to you, but that's what I generally carry along. And then I pretty much have everything that I need, depending on what I find. So, any questions then about equipment for collecting? Of course, you need a field guide, and I'll say more about that in a moment. Okay, so I'll say more about the field guides as part of the presentation, the PowerPoint presentation, but I'm going to leave them for a moment. I will say one thing. I don't know how many of you are not members of the local club, the Eastern Penn Mushroomers. Is everybody here? Okay, there are quite a few. If any of you are interested in joining the club, and that's of course totally up to you, I did bring some little flyers about the club, and if you are interested, you can join up. The membership is $15 a year. We go out about every other weekend in the summer, and we also have 3 meetings here in this room in the winter. It's a pretty active group, and certainly a very friendly group, and we try to go out, and basically our procedure is we spend about 2 hours in the woods, and then we gather and we lay out all the things we found on a table, and we try to sort them and identify what we have. And of course, if there are some edibles, that's great. And then actually we do a pretty good job of sharing those among the people. And once a year we have a tasting where we get together and we try these wild mushroom dishes. And then we also have a multi-day foray, which is held over in the Michaud State Forest, and that's going to be coming up this July. So we stay at the Mont Alto dorms, and we go to several of the state parks and forest areas there. Anyway,
John?
Yeah.
Another thing that's really wonderful about the winter meetings is that they have really interesting speakers on specialty areas, but there's all manner of treats that the members bring for that.
Yeah, this is the only meeting actually in which we don't have treats here today. But usually we have a very good culinary coordinator who saves up mushrooms that he's found during the year, and he has quite a larder that he draws upon, and he loves to cook, and he and his wife really bring some great stuff.
How does he preserve those then?
How do you preserve the mushrooms for use?
Yeah,
Depends on the species. The easiest way is to dry them, but there are some mushrooms that don't dry well. The flavor is lost. For example, chanterelles. For chanterelles, what we do, we simply saute them in some oil or butter and then freeze them. And for most mushrooms, that's a good way to do it. It's a little more time consuming perhaps than the drying technique, but it does, they're a little bulkier that way. When you dry them, of course, they shrink greatly in size, and then you reconstitute them with water. So for things like morels dry very well. But chanterelles are an example of one that really, for some reason, loses the taste when you dry. So yeah, there's various options of what you can do. Other questions before we start the presentation? Okay, Judy, if you can hit the down arrow here. So I know what it's like when you first start out. It can really seem overwhelming trying to figure out what mushroom you have. And at first you may think, well, I just don't have the right book. Maybe if I had the right book, it would be in here. The problem is, in Pennsylvania alone, it's estimated there's about 8,000 described species of fungi. And it's thought that that's about 10% of what's actually out there. So there's a vast number of things out there that have never been described. Now, a lot of them are microfungi. They can really only be seen with a microscope. But even setting aside those, there's still a lot of mushrooms out there that nobody's yet had the time to name and describe. So there's no book that's going to have them all, and you have to realize that right at the beginning. But most important, especially if you're interested in eating wild mushrooms, the thing that's most important to understand is there's no simple way, just by looking at a mushroom, that you can tell at a glance whether it's edible. All these old wives' tales about cooking it with a silver spoon or various other instantaneous sort of methods, they're false and they're very dangerous. So the only way to do it involves a considerable investment of time and effort. And you're going to have to observe not just one feature, but combinations of features. So let's go to the next slide and see what basically you're getting into. You are going to find that you're going to need to study field guides and other literature. Hopefully that will be an enjoyable task. I mean, for me, I can sit down and just get absorbed in some of these things, but it depends on your interest. As I just said, it's really important to observe combinations of features. So, for example, if you look at the Peterson Field Guide to Birds, that works very well for birds because you can have one or two little arrows that point to critical features, and if the bird has those markings, then you know what it is. If you've ever looked in the Peterson Guide to Mushrooms, which you may not well have seen, it's not available in most places, it attempts to do the same thing for mushrooms, and it's a failure. It just doesn't work well because there's not just one or two things that you look at and identify the mushroom. It takes a lot more study and a lot more combinations of things than that. So that pointer method that the Peterson Field Guides use is great for birds, it's great for wildflowers, but it doesn't work for mushrooms. Then when you have a field guide and you start to use it, you have to make sure that when you look at the mushroom, that it really exhibits what the guide is saying. Because the guide can kind of lead you on. You can say, oh, well, yeah, I guess maybe I'd say that those gills don't touch the stalk. In other words, when you read the description, it sort of suggests something to you, and you have to resist taking that suggestion unless the mushroom really does exhibit that character. You also have to be willing to admit you've made an error. So you may work down through a key and suddenly you're stuck. Well, either you made a mistake somewhere earlier, or else maybe that particular mushroom just isn't in the book and you're at the end of the search. But you have to be willing to admit that you might have made a mistake in some of your judgment. And finally, you do have to have a high tolerance for frustration, because there will be a lot of mushrooms that you find that you just don't succeed in keying out. And it may be because you're inexperienced, it may be because the key's poorly designed, it may be that book doesn't have that particular specimen, or it may be you found one of these 90% that simply hasn't been described. So you do have to have a high tolerance for frustration. Okay, now I think that you can sort of classify mushroom collectors in about 4 groups. So as a beginner, you have to depend on other people's identification skills. Well, that's fine unless you're planning to eat the mushrooms, and then of course you have the question, who do you trust? You have to have some reason to think that the person that tells you this is an edible mushroom knows what they're talking about. And so that's a matter that you have to decide on your own. When you get a little more experience and become what I call an apprentice collector, you're able to identify the really poisonous mushrooms, so you're over that hump. You know some of the good edibles and a few other of the real common species, but you're not very good at the use of identification keys. So if you find more than those handful of species, you're still having trouble identifying things. When you develop a lot more experience, then you're able to identify a lot of species without using field guides. You know what to look for in terms of identification features, and you've become proficient in using keys, and you know what some of these technical mycological terms mean. I'll talk more about some of these specialized terms that you will find in any field guide, and you do need to know a few of these in order to efficiently read the guides. And then finally, unfortunately, some people that have a lot of experience get a little complacent and careless and just don't spend as much time maybe as they should, and that's dangerous. So I mention that because anybody can make a mistake if they get careless. And with something like poisonous mushrooms, that's a risky thing. So you always have to be really careful, especially if you're going to eat the mushroom, to check what you've done, to check you're going through the key, hopefully check with somebody else that's equally skilled, to just make sure that you haven't made some mistake through carelessness. Okay, when you collect mushrooms, it's very important to segregate your different collections that you've made in different places into separate bags. Okay, so you don't want to just lump everything in your basket together. You don't want to put more than one collection in the same wax paper bag, because you might have two different things, and if you put them all in the same bag and then just take one or two out to identify, well, you may correctly identify those, but if there's some other things in there that are not the same, then you've got a problem. It's very important to collect the whole mushroom. And if you can, if there's more than one, try to get a young one as well as a mature one, so that you can observe things like color changes. But the point of getting the whole mushroom is that sometimes there's some things under the ground that you don't see at first glance, and sometimes those are critical identification features. For example, there are some mushrooms that are parasitic on pupae of insects. And if you just pick off the mushroom and you don't dig down, you'll never realize that there's an insect pupa there. And once you see that, it's an immediate identification almost of what the genus of mushroom is. As I indicated, you should have some sort of slip, or at least a pad of paper, that you can record some of this ephemeral data and just toss that slip in the wax paper bag with the mushrooms, so that you have that information at your disposal when you get back and you start using a key. Then, really important is this one on trying to get spore prints. So what's a spore print? If you take the cap of a mushroom, I'm talking now about the standard parasol type mushroom, take the cap and put the underside face down on a piece of white paper, or even better if you happen to have one along, a piece of glass like a microscope slide. And protect it, so put it in your little plastic bag or take it home and do this, and maybe cover it so that there's not a lot of air currents floating around. If it's a fairly fresh mushroom, within about half an hour or so, you should start to get these spores, which are the reproductive elements of fungi, to deposit in large numbers on that paper or that glass. And what you're looking for is the color of those spores in mass. That is usually the first step in keying out what a mushroom is. And the reason is that there's only a limited number of colors you can get. It's an easy thing, usually, to judge. So that segregates or separates the mushrooms right at first and eliminates a lot of possibilities. And the other thing is that unlike the cap and stem of the mushroom itself, the color of the spores does not change as the mushroom grows older. It stays the same color. Color is a very bad thing to use for judging what something is if you're talking about the cap color, because that will change a lot depending on whether it's dry, wet, young, old. There's a lot of possibilities. But the spores, the color does not change. And in a number of important cases, there are lookalikes, one of which is poisonous and one of which is edible, and they can be distinguished by the color of the spores. So taking a spore print is really a critical thing. Now, unfortunately, if you have an older specimen, you may not get a spore print. So you can't always do that. Sometimes you can judge the spore color because some spores will have actually fallen down on the stalk, and you can look at that. And if worst comes to worst, you can look at the gills or pores from which the spores have come, and they may be stained by the spores. But that's a little iffy because it may not be mature enough to have started to deposit spores, and in such a case, it may not have stained the gills either. So let's go to the next slide, which gives an example of what a spore print looks like. So this is an example of one that has a light brown spore print. And this is from a gilled mushroom, one that has those parallel sort of plates that bear the spores. So you have this sort of projection onto the piece of paper of this pattern of the gills. And you can not only tell the color from that and the fact that it is a gilled mushroom, but you can also judge the spacing of the gills, which is something else that can be useful. Okay. So if you're lucky, you'll get something like that, and then you look at that color, and that's the first thing that your key will probably ask. Are you talking about a mushroom with white spores, brown spores, pink spores, black spores? Well, in this case, you'd say, okay, I've got light brown spores. Just a question that all of you should be able to answer. If you go out and you buy a button mushroom in the grocery store, just an ordinary white button mushroom, what color are the spores? What happens when you handle it in cooking? What happens to your hands? They get discolored, right? Well, your hands, of course, are pink, but if you lay it on a piece of white paper, you'll find that it's actually a chocolate brown. Your hands may look sort of black because of the normal pinkness of your skin, but they're chocolate brown. And one of the major genera of mushrooms is called Agaricus, and the general term agaric, A-G-A-R-I-C, simply means a gilled mushroom. Anyway, Agaricus is a genus of mushrooms to which that commercial mushroom belongs, and one of their characteristics is that they have chocolate brown spore prints. So this is not an Agaricus, this is not chocolate brown, this is too light to be that. But if you take a grocery store mushroom and just practice making a spore print, you can get an idea of what some of these colors are actually like, because it's hard to describe colors in words sometimes. Okay, let's go on. Now it's easy to get a field guide and start looking through pictures, and that can be very helpful. But you shouldn't rely on pictures alone. You should look at the verbal descriptions as well, and again, in conjunction with keys to help you narrow your search. The reason you can't depend on either one, well, colors, as I just said, are difficult to describe, and they're also difficult, some of them, to reproduce in photos. In particular, there's a sort of a magenta color that I have never succeeded in rendering correctly, either on film or in digital camera. There's just something about that shade that's very hard to do. And again, the colors of the mushroom itself vary at different stages of growth, so they're not going to remain the same. And once again, that's why spore colors are what you want to focus on. Now I want to talk about keys, and so I want you to refer to your little handout that I gave you. I'll talk more about the first page in a moment, but I want to talk about various types of keys. So if you look at the second page of your handout, you'll find an example of the pictorial key. I'll go over that more in just a moment. In this sort of key, the mushrooms are arranged by basic shapes or what we call statures. So these are stature types. So you'll see this first column now. Don't be put off by that word basidiomycete. I'll explain in a moment what that's all about. But you can see some mushroom shapes, and you can see they differ. So the first one is kind of almost, one of them is almost funnel shaped, and it has these gills that are sort of going downward on the stalk. You've got another one down below it that's also funnel shaped, but doesn't seem to have much in the way of gills. Then you've got the more or less standard parasol shape. You've got one that's growing on the side of a tree like a trunk, or with a stalk maybe, but it's off to the side, it's lateral, and so on. So just by looking at the shapes, you can start to classify what you found. So that's an example of a pictorial key. And this one is from this book called Mushrooms Demystified, which is one of the books that I highly recommend. It's not something you can carry in the field, it's too heavy, but there is a mine of information in this book, and it's by far the most comprehensive guide to mushrooms that exists right now. You can get it from Amazon. You can get all of these basically from Amazon, and the price on that one, a new price is $26.39, and used you can get it for $19.99. It's an extremely good book. I do recommend that. So this pictorial key is from there. So is the next one, which is also a type of pictorial key, but I prefer to call it a tabular key because it's in table form. So again, it does have pictures along the left side there. And again, it's the shapes and the differences in those shapes. So you look for that, and once you've found the shape, then you go across the table, and the columns represent, for example, spore color. So in the one column there, the spore print is white to buff. In another one, it's pinkish. The third or fourth one over, I guess, is brown to rusty brown, and then the last one is black. So this combines shape and spore color, and then within each little cell there are a list of the names of the genera that it could be. So again, we're narrowing the search. We really need to know what species we have. This is a first step, finding out what genus it belongs to, and then you can go to your guides, look within that genus, and get more detailed keys to the individual species in that genus. Do I need to say anything about how biologists classify things? When I use the term genus and species, does everyone basically know what I'm talking about here? And when you break things down, you start out with kingdoms, and fungi basically form one whole kingdom of organisms, just like animals do and plants do. Now then you have smaller classes. You have phyla, you have what are called classes, you have orders, families, genera, and finally species. So you work your way down. Well, we're skipping almost all of those down to the genus level by using a key like this. So normally you're not going to have to worry about the class and the order and even the family. Okay, so we've seen examples of two types of keys, the strictly pictorial key and then this tabular key. Then there are keys that offer you numbered choices. Let's see, let me find, okay, this third page gives you a simple example. If you find a mushroom that doesn't have gills, you can start to put it in one of these various groups. So for example, maybe the fungus you have looks sort of like a coral. Well, within that, there's 5 choices there, okay? And so depending on which of those choices seems to fit, you have the name of the genus over there on the right. Okay, so this one involves numbers. Now, many numbered keys will have a lot more detail to their choices, and in particular, many of them will give you just 2 choices. That you'll make a series of choices from 2 alternatives. Those are called dichotomous keys. So at step 1, you make a choice, maybe as to spore color. At step 2, you make a choice as to how the gills are attached. And then at step 3, you make some other choice. As long as there's just 2 choices each time, it's called a dichotomous key. On the other hand, if there's more than 2 choices, at least some of the time, then it's a non-dichotomous key, and those are a little bit more work to use because you have to kind of hold things in abeyance while you read all the various possibilities. So a dichotomous key is easier to use, although it may take you longer, may take you more steps before you finally end up with an answer. Most people prefer dichotomous keys if they're going to use a textual type key. Now, one disadvantage of detailed textual keys is that you go through these various choices, and at some point you realize you're heading toward a dead end. None of the choices that you reach seem to apply to what you have. So that means you've got to backtrack. But an awful lot of textual keys don't tell you where you came from. So they will have some numbered choices, but say you get down to step 5, you look at both choices in step 5 and neither one of them fits. Well, now you have to go back and you say, well, how did I get to step 5? Where did that come from? Well, unless the key is designed some way with indentation or with some reference numbers, you have trouble going backwards to see where you might have made a mistake in one of the alternatives. That's where flowcharts come in. And so I've given you a bunch of flowcharts here, which are the ones that I tend to use in the field. These are basically based on a textual key that's in this book, about which I'll say more in a moment. But it's been rendered in pictorial form. And this was basically prepared by a woman in the New Jersey Mycological Society. And so the simplest of these is this flowchart for pink-spored agarics. So again, an agaric is a gilled mushroom, and some of them have pink spores. Relatively few have pink spores, so that means that this is a relatively simple flowchart. And it is dichotomous because at each node here, you just have 2 choices. So suppose you take this mushroom you found, you make a spore print, and it comes out pink. All right, then you start with the first choice. Does it have a stalk or not? So the choices here are no stalk and growing on wood versus having a central stalk. You make one of those choices. If it happens that it doesn't have a stalk and it was growing on wood, then you're done with this key. It says, well, then you probably have something in the genus Phylotopsis. If it has a central stalk, however, then you've got further choices. So now you have to look at the gills and how they're attached, and I'll talk more about those terms in a moment. As you go down further, again, there's more technical terms referring to the gills, referring to something called a volva, about which I'll say more. It's not a Swedish car. It's something that has to do specifically with mushrooms. Then the size of the caps, you can see that choice further down, and so on. So you gradually make your way through this chart. But you do have to know some technical terminology to get through it. Okay, that's a relatively simple flowchart. Well, if you have dark-spored gilled mushrooms, so that's basically black, purple-brown, chocolate-brown, and so on, then you still have just a single page here, so it's still not too bad, and it's very similar to what we were just talking about. On the other hand, if you have a light brown spored, well, then you still don't have too much trouble, except that you're having to decide, well, is this really light brown or is it a dark brown? Because if it's dark brown, I should have used the other page. If you're convinced it's light brown, then you would use this one, and again, make your choices. Now, unfortunately, I should say, this one has 2 sides, unlike the others, okay? So this one is a little more complicated. So there's more brown-spored mushrooms than there are the dark-spored or the pink. If you have a light-spored one, white or light buff, then you've got a problem, because this one's stapled, and there are 5 pages of choices here. There's lots of white-spored mushrooms out there. By the way, you might say, well, how do I know if it's white-spored? Because I'm taking this print on a white sheet of paper. Well, if you hold that paper up at a slight angle, you'll still be able to see the spores. There will be that radial pattern of lines. And even though they're white, on white, you'll still be able to see them. It looks sort of like the paper's been embossed in a pattern of ridges. So you can still see them. However, that's one good reason actually for using a microscope slide rather than a piece of white paper, is because if you've got a clear microscope slide, then you can see it no matter what color it is without any trouble. Okay, so light-spored ones are the ones that are going to cause you the most trouble, and there's a lot more genera. Now, it's possible that you have something that doesn't have any of these colors. These cover most of the possibilities, but there are a very few, actually I can only think of one genus offhand, that have green spores. If it has green spores, that's a winner. because you can tell what it is right away. There's not much other choice. It's also important because that green-spored mushroom is common and it's really poisonous. And so if you get one that has a green spore print, you've got a chlorophyllum, which basically means green gills, and it's one to avoid. We'll talk more about that one in a moment. So let's go through the next few slides and just reproduce these. Okay, again, these first two are both from this book, Mushrooms Demystified. So you have a choice in that book of whether you start out just using this key that's primarily on shape and does not make reference to spore colors, or once you've sort of gotten good at using that key, or at least as a first step, this breaks them down into some sort of large groups of types of fungi. You can go on to the tabular key, which we've already seen, so next slide. This one is a little more detailed because now it's not just telling you a large group of mushrooms, but it's actually giving you a list of genera. So this key is much more detailed, but it's still got the pictures there to help you. And you have to look, though, more closely at the pictures to see what the differences are. And that's what this first column of characteristics is all about. It's a verbal description that goes with the picture to help you distinguish, for example, picture number 5 from picture number 6, which look rather similar at first glance. Okay? All right, then this is another example of a dichotomous key. I don't think I put this in your handout. This is what a textual dichotomous key looks like. So you always have two choices. You start with number 1, there's 2 possibilities. You make a choice, you go over, and then it tells you, okay, if you made this choice, go to such and such. Either another place in the key, that's what the numbers are, or a specific page in the book and a specific group of mushrooms. And you hope, of course, that eventually you come out to a specific group of mushrooms, then you go to that page and you'll probably find another key that tells you within that family or within that genus what more to look for. Well, you can already see it's going to be a lot of work. I mean, it's time consuming going through these keys. And there will be a number of places you'll hesitate to make a choice. You may not be sure. And so that's why you have to be aware that you may make an incorrect choice and you may have to backtrack. With this kind of key, where there's no indentation and there's no numbers that tell you where you came from, you may have a lot of trouble backtracking. So, let's suppose you're down here at number 16. You've gotten that far, and for some reason you have trouble there, and you say, no, I think I must have made a mistake somewhere. Well, how'd you get to number 16? Well, you have to do a random search over here to discover, oh, I got to 16 from choice 10. But that takes time to find out where you came from. And if you have a really long key, you may actually have to page through to find out where you came from. So these kind of keys are most useful if they give you some kind of cross-reference that allows you to backtrack. You don't have that problem with a flowchart because you can see immediately what the path was, how you got there. So they're much easier to backtrack if you are hesitant about a choice or you made a mistake. Okay, again, we've seen this as an example of a flowchart key, so I don't think we need to say any more about that one. So what strategy should you adopt? Well, I suggest you start out by learning these major morphological groups. And of course, morphological just means the form, the gross form of the thing. That's what that first key really sort of did, that first pictorial key. It more or less broke the mushrooms down into these major groups. So you could use that one for that purpose. You could just look at those general shapes, and that pretty much tells you what those groups are. And we'll go through those in more detail in a moment. But this kind of thing is what I'm talking about when I'm referring to the morphological groups. It's important not to get overwhelmed, so concentrate on just learning a few species at a time. Don't expect to go out and identify very many of the specimens you've collected. So there's a temptation to collect everything in sight. Well, what happens is you get home and maybe you identify 2 of the things you got, and the rest you have no idea what they are, and you just feel really frustrated. So it's better to work on a few species and gradually expand your repertoire. I strongly suggest you learn the most dangerous ones first, and especially those that have lookalikes that are edible. I listed some examples. I'll show you pictures of these later on in the presentation. And the other thing, as you've seen already, you have to make your peace with Latin names. There's no way around it because most mushrooms do not have common names at all. So the only way you're going to be able to refer to them is to, roll up your sleeves and get grubby with the Latin. It helps if you have a book that gives you a little bit of glossary. And that's another nice thing about this book. In the back, it has a list of Latin prefixes and combining forms so that you can sort of translate some of these Latin names, and that makes it easier to remember. For example, you find this puffball, and you find that it's Latin, the name of the genus is Lycoperdon. Well, that's not particularly easy to remember. But if you look it up and you discover that that means wolf fart, you're not likely to forget it, right? Now, how did it come up with that name? Well, Linnaeus named it. Maybe he got awfully close to a Swedish wolf, I don't know, but it's referring to the puffing sound that it makes when it puts its spores into the air. So some of these names are actually humorous if you translate them. And there's other things, like a lot of genera end up CYBE is a Latin combining form. So you have Clitocybe, for example, Inocybe, and basically CYBE means cap. So it's telling you something about the cap. Clitocybe means it's got a sloped cap. Well, if you know a few of these, then you can figure out what they mean. Again, chlorophyllum means green, as in chlorophyll, and phylum is the Latin word for gills. Okay, so literally phylum is a leaf in the sense of something flat, like a page that's a leaf in a book. So phylum literally means that, and in the case of a mushroom, what's flat are these surfaces on which the spores are born. And same in a fish gill, it's the same sort of structure. Okay, and sometimes you can tell right away. I mean, scleroderma, you can almost translate that from what you know. Sclerosis, multiple sclerosis, what does that refer to? Sclero, does anybody know what that means? Pardon?
Isn't that hardening?
Yes, exactly, right. You can talk about arteries, arteriosclerosis, right? Hardening. Derma, hard skin. So that's what scleroderma is. It's something that looks like a puffball but has a hard skin. Okay? And citrina means yellow. Okay? So, with a little knowledge, the Latin names aren't so bad. Now, one thing that can be confusing, most of us in the field use this Audubon Society field guide, and I'll talk more about its pros and cons. Unfortunately, the author, who's very knowledgeable mycologist, was forced by the publisher to give common names, that is, to make up common names for everything in the book. So you will look in here and there'll be some common name given, but nobody but this book uses that in most cases. So I think that's very unfortunate that the publisher insisted on that, because the Latin names are in universal use. But most of the common names in here are unique to this book. So don't use them because other people are not going to know what you're talking about. And many times they're actually just a translation of the Latin anyway. But okay, so once you've learned the really deadly ones, then you go on. I think we skipped one there. I think it was on for just a minute. Yeah. Then you go on and learn the safest edible species. And that means those that are least likely to be confused with something that's toxic. So the familiar edibles, morels, chanterelles, giant puffballs, you're not very likely to make a mistake. There's nothing else that looks like a giant puffball. I mean, this is a huge round thing, maybe softball or basketball size, and it's growing on the ground with no stalk, usually has a very smooth skin. You're not going to mistake that for anything else. Morels also are pitted, they're pretty unique. Now chanterelles, I think, are pretty easy to tell. Some people do make a mistake, and I will talk more about what they mistake it for. But certainly morels and giant puffballs are very safe, and for most people I think chanterelles are as well. Now there's other edible species, so-called honey mushrooms, Armillaria mellia. The winter mushroom, Flammulina velutipes, which is also one that's actually sold in the grocery stores. Some of these others are good edibles, but leave them to more experienced collectors because they do have lookalikes, and some of them can be dangerous. So don't try these until you've really developed some experience. Yeah?
John, is there a mushroom that's so poisonous that you could actually absorb the toxin through your skin?
No, you don't have to worry about that. Now, if you do handle one of these really toxic ones, I would certainly wash your hands before you eat something, because even the spores are poisonous, actually. Now, it's not too likely that you would get enough on your hands. On the other hand, we'll see the so-called destroying angel, which is one of the amanitas. Toxicologists measure toxicity in what's called a number that's called the LD50. I don't know whether you've ever heard of that. It stands for lethal dose for 50% of the population. It's a statistical measure, which means that 50% of people, if given this dose, would die, okay? Because it does vary, from person to person what would be a fatal dose. And mostly it depends on your weight. So these LD50 numbers are given as so many milligrams per kilogram. So on this amanita, the LD50 for that amanita is 0.5 milligram per kilogram of body weight. Now let's think what that means. A kilogram is about 2 2/10 pounds. So 100 kilograms would be a 220-pound man, say, okay? So if you weigh 100 kilograms, or 220 pounds, take that 1/2, multiply it by 100, that says 50 milligrams would be a lethal dose for half the population at that weight. Now, how much is 50 milligrams? Well, I take a Lipitor pill that's 20 milligrams. It's a small pill. So 2.5 of those would weigh 50 milligrams. That's not much. That's certainly less than 1 cap, a lot less than 1 cap. So that's really toxic. That 0.5 milligram per kilogram is more toxic than cyanide, volume for volume. So we're talking about something that's really poisonous. So I'd wash my hands. I'd be real careful about that.
Are they using any of these toxins in research for medicine?
Yes, actually. In fact, the one that I was just talking about. It turns out the way it kills you, it inhibits an enzyme you have called RNA polymerase, which your body uses to make RNA. And when you absorb this chemical, your body can no longer make RNA. And while that's very bad for you, in particular, it's going to put you on the liver transplant list right away, it's useful for geneticists because it's one of the few chemicals known that will stop RNA at a specific point. And so it is actually used. In fact, one of the chemists on our campus told me he once used some of this stuff in a genetics experiment. He said it was absolutely the most hazardous chemical he ever handled. And it came in a big box. And he opened that box, and within it is another box with a warning, at this point you should be wearing this. And this went on about 6 layers, each layer telling you at this point you should have put on this, and finally you ended up with a full hazard suit by the time you got to this small bottle of concentrated stuff. So yeah, some of them are useful. Another one of the toxins, and I'll show you the picture of that one too, was actually identified as a result of the space program. Because it turned out somebody in a rocket fuel plant got sick from, I guess, fumes. And somebody who worked there was a mycologist or mushroomer and recognized that the symptoms sounded similar to what he'd read about this one false morel. And they tested the mushroom, and sure enough, it's a natural source of this rocket fuel oxidant. So that's another case where the chemical is useful, but I mean, it has nothing to do with mushrooms per se, but that's how they actually identified what it was. Okay, so let's go on then, once you've made that decision. Also, there are some groups of fungi that are just really hard, and definitely leave them to the experts. That's true of almost any area of nature study. For example, bird watching, fall warblers. You know, if you're not an expert, you don't try to figure out what that fall warbler is, because they all look so much alike. If you're interested in wildflowers, you kind of grown when you have a so-called DYC, damned yellow compositae. It's one of these things like a sunflower, you know, that has radiating yellow petals. Well, there's jillions of them. Well, for mycologists, it's the LBMs, the little brown mushrooms. There are a tremendous number of small, nondescript, little brown mushrooms. They are very hard to tell apart, and one or two of them are deadly poisonous. So leave those alone. You're going to be terribly frustrated if you try to identify them, and if you make a mistake, especially if you eat one, you may be in big trouble. There's a lot of fungi that are important in the woods. They digest wood and leaves, but primarily wood, and they just form flat crusts on fallen logs, for example. They can be interesting, but most field guides don't give very good coverage of them. So, although you'd like to know what they are, you may find that it's pretty frustrating, because there are a great many of them and they're not that easy to distinguish. There's a genus called Eniloma that has one edible species, but again, if you find one of these and you discover from one of your keys that it's something in that genus, you're in for a really tough time, and you probably would have to resort to a microscope or chemical test or something like that to get the species. And there's another genus called Mycena, also lots of Mycenas, and a real challenge to identify those. Well, then finally, you reach this point. Do I eat this thing or not? And that's a decision only you can make. You're the only one who can decide when you're confident enough that you've correctly identified this, that you're willing to risk ingesting it. Until then, you definitely need to check the identities and make sure that, you know, they're confirmed by someone whose skills you trust. That can be a real test of friendship, whether you trust somebody. Not that you think somebody's intentionally trying to poison you, but you just think that maybe somebody doesn't know as much as they think they do. Okay, so let's talk a little more about these major morphological groups. So the first group are the ones that have gills. Basically, mushrooms, scientifically, are classified by the structures on which the reproductive elements, the spores, are born. So the most familiar mushrooms have these parallel or nearly parallel plates that we call gills. The spores are born on the sides of those gills, and when the spores are ripe, they are ejected from the surface of the gill. Now, there's not too much space between the gills, but spores are very, very tiny things. A typical spore is about 10 microns in its long dimension, and a micron is a millionth of a meter, or a thousandth of a millimeter. So 10 thousandths or 1/100th of a millimeter is the long dimension on a typical spore. That's way too small to be seen with the naked eye. And even with a scope, to see them well, you need about 400 power to get a good view of a spore. So anyway, these spores are very tiny. They're ejected into the space between the gills. They fall and are carried away by air currents. Now, in some of the more evolved fungi, those gills have developed cross veins, and so now you have pores, because you don't just have plates going this way, you've got plates at an angle to them, and so now you've got these little cells that form pores. There's a greater surface area, so they can bear more spores. And there's two groups. There's these fleshy ones that are soft, and that usually have central stalks and still have a typical parasol shape. Those are called boletes. And then there are these woody, very hard ones that grow like shelves on trees that have pores, and they're called polypores, which just means many pores. So the differences are the texture, woody versus fleshy, and also the pores on the polypores are much finer, much smaller than they are on the boletes. The pores on boletes are visible to the naked eye. Some of the ones on polypores are not. You have to use a hand lens to see them. Less common but interesting are what are called hydnums. They have icicle-like teeth hanging down from the cap, and they bear their spores on those teeth. Then we have the morels, true and false, which have pitted surfaces if they're a true morel, or a convoluted sort of brain-like wrinkled surface if they're a false morel. Chanterelles, which are usually funnel-shaped, and they may look like they have gills at first, but if you look closely, you'll discover they're just blunt ridges. They're not really gills. They don't have much depth to them. They're just sort of very, very shallow ridges. So those are the chanterelles. Okay. Then there are these strange things called club fungi that look like little fingers sticking out. So some of them are called earth tongues. And there is this genus Cordyceps, which are the parasites on insects. I mentioned one of those earlier. They have these finger-like growths coming out that bear the spores. There's a large group of fungi called coral fungi that look like coral reefs. I mean, they have the shapes of corals. There's another large group that are very gelatinous in texture. If you like Oriental food, and in particular, if you like hot and sour soup, there is that kind of cartilaginous stuff in hot and sour soup. It's kind of crunchy, doesn't have much taste, but it's a little flabby. Well, that's an example of a jelly fungus, an edible jelly fungus. Then there's a group called gastromycetes, literally means stomach fungi, whose spores are enclosed inside the fruiting body, and they include puffballs, which expel their spores by forming a little pore, and then anytime a current of air or maybe a raindrop or something hits, that thing puffs a bunch of spores out. There are earth stars, which are very similar to puffballs, except they start out and they're kind of wrapped in an outer coating, and that breaks into petal-like things that open up, and then there's a puffball inside. Stinkhorns, which you probably have encountered in your gardens, which smell absolutely terrible. Most of them are sort of phallic in shape, and they have this green slime at the tip that smells like rotten meat or whatever. They distribute their spores not by air currents, but by insects that land on this smelly stuff, thinking they found some rotten meat or whatever, and they carry off the spores from this sticky stuff. Bird's nest fungi, which are also very common in your garden mulch. You probably have some and may not even have noticed it. They look like tiny miniature bird's nests, complete with eggs. And it turns out those things that look like eggs contain the spores, and they depend on raindrops. Hitting this nest-like structure knocks these things out, and they've got a cord underneath, so when they get knocked out, this cord elongates, and they've got a sticky pad on the end, and if they come in contact with a plant, that pad sticks, and then this thing that looks like an egg hangs from it, eventually disintegrates, and the spores fall on the ground. And truffles are another example. How do truffles distribute their spores? Because they grow totally underground. Well, they have very strong odors, and they depend on animals to find them and eat them. They pass through the animal's digestive tract and then are deposited in the feces of the animal. So all truffles, for example, reproduce in that way. That's why they use pigs to hunt truffles, because they use sows to hunt the truffles, because it turns out that species of truffle, the odor is exactly the same as a sexual pheromone that a boar produces. And so these sows, when they smell one of these, they think they found a real stud out there. But actually they found a truffle. And so they also use dogs. The claim is that, like cadaver dogs, truffle dogs don't really like the smell of truffles, but you can train them to find them. And that's actually good, because unlike the pigs, they don't eat them. So the dog will find the truffle for you, but just leave it. There's also lots of fungi out there that look like miniature cups. Some of them you may even find growing on your carpet. I have a neighbor who a couple of weeks ago came to me in distress. She had one of these things coming up through her basement carpet. They'll grow on lots of different things. And then you have these crust fungi that I mentioned before that just are kind of flat on the surface of fallen logs. So that's an example of some of these many major morphological groups of fungi. And you can at least group things to that extent.
Okay, the truffles grow underground.
Yeah.
But the spores then get deposited on top in the feces, so the spores just gravitate.
So eventually the spores will get on the surface of the ground and then will germinate to form a mycelium, which is the network of tiny microscopic threads that are really the vegetative part of a fungus. So when we're talking about a mushroom, we're talking about the fruit of this organism. But with any mushroom, there is this very extensive network of microscopic threads that are gathering the nutrients. And what those threads do, they actually exude digestive enzymes. So one of the differences between fungi, for example, and animals, our digestion occurs inside our bodies. With fungi, the digestion occurs outside their bodies, and then they absorb this fluid that has digested the nutrients. They absorb it back into these little filaments. So the digestion takes place outside the fungal body. Okay, so now let's talk about what you should observe. First, you want to observe the size. So, if necessary, take a little ruler along. Actually, many field guides even have a built-in ruler. So I think this one, for example, has a ruler on the back cover. Now this one's a little bulky to carry along, but lots of field guides will have something like that. So you can measure how broad is the cap, how long is the stalk, and so on. When it really gets tricky, when you get one that you have to go to a microscope, you may have to measure the spores, and that requires some special stuff. You have to have about 1,000 power lens, and you have to have a device called a reticle, and that's getting fairly technical. It even gets worse than that in some cases. Some spores have warts on them, and there are even people who measure how big the warts are. You really got to be hard bitten to reach that level. Okay, then the shape of the fruiting body. We've already seen a lot of varieties here. The nature of the spore-bearing surface, whether it's pores, gills, teeth, whatever. The color of the spores, the color of the gills, and also the color of the cap and stalk, which may vary. The spacing and arrangement of the gills or pores, and the shape of the pores. All of those can be critical, and there are a lot of technical terms that refer to how the gills are attached to the stalk. So if we can go to the next slide here. Some mushrooms of these parasol type that have gills, the gills don't reach the stalk at all. They simply go up into the cap before they get to the stalk. And as a result, you have what looks like a sort of a clear moat around the stalk. Those are referred to as free gills. Otherwise, they're attached gills, meaning that the gills do reach the stalk. Okay? And there's various ways they can be attached. You want to look at the texture of the cap and stalk. So, is it slimy, for example? Is it powdery? Is it waxy? There's one whole group that feel like candle wax when you handle them. Are there distinctive markings or some sort of ornamentation on either the cap or the stalk? And in particular, on the cap, do you see any sort of cottage cheese-like warts or any scales that look like kind of lifted up, like you could peel them off? Are there evidence of kind of a veil that hangs off the edge of the cap? Or is there something on the stalk that looks like a skirt? Or maybe a cup at the base of the stalk? All of those are very important things to observe. Now sometimes some mushrooms actually exude drops of liquid, and the color of that can be worth noting. And especially among the boletes, many of them, when you handle them or when you cut one, they suddenly change color. In particular, many of them turn blue. That's very important to notice. There are some other possible color changes, but that's the primary one. And does the specimen have a distinctive odor? Unfortunately, we don't seem to be very good at describing odors. And what smells like watermelon to one person may smell like something totally different to another person. So when you read descriptions in books of odors, you kind of have to take it with a grain of salt. There is one edible mushroom out there that's very distinctive. It's large, has scales. I think it smells exactly like watermelon, but other people find other smells that they think it smells like. So note if it's distinctive, but you may get in arguments with other people as to what the smell is. And there are some terms that are used in mushroom books. For example, it'll say it has a farinaceous odor. Well, you look up that word in a glossary and it's supposedly the odor of new meal. Well, if you're not a miller, I don't know, to this day, I don't know what new meal smells like. But I guess go to your local mill, if you can find one that still grinds grain, and take a whiff. Anyway, that word is used a lot. So let's talk about some of these terms that you're going to encounter. The first two refer to microscopic characteristics. They're not something you're going to observe, but you will have to know these words just to use the field guide. Because you noticed in that one group, on that very first key, it had that term basidiomycetes. Okay? So in other words, this key only applies to that group. Well, many of your agarics, many of your guild mushrooms, the spores are born on these microscopic cells that are club-shaped. And they have four little prongs, usually four little prongs called sterigmata on which the spores are held. And at a certain point, they're expelled from those one at a time. Anyway, those club-shaped cells with these prongs on them are called basidia. So any fungus in which the spores are born on basidia is called a basidiomycete. So this is a class. It's at the class level of classification. On the other hand, most of the other fungi you're likely to encounter are ascomycetes. So morels, for example, are ascomycetes. And in those fungi, the spores are contained inside microscopic sacs called asci. And at some point, the tip of the ascus, that's the singular, the tip of the ascus develops a hole. There's more than one way that can happen, but at any rate, if the spores are born inside these sacs, then it's an ascomycete. Okay? So that's the meaning of those terms. Again, without a microscope, you're not going to see these things. And it takes a little practice especially with the basidiomycetes, to see them even with a scope. But anyway, that's the real technical way of classifying fungi at one of the higher levels. You'll see the word pileus. That's a fancy Latin word that just refers to the cap. And likewise, stipe is the technical term for a stalk. You might say, why don't they use cap and stalk? Well, informally, most of us do say cap and stalk, but I guess technically a stalk refers to something like a plant where you have vascular elements, you have what amounts to veins and things like that, and mushrooms don't have that. Okay, annulus literally means a circular band, and in the mushroom terminology, it means a ring or skirt on the stalk of the mushroom. And it's one of the more important characters to look for. And this word volva that we saw before means a cup at the base of the stalk, which may be open and not touch the stalk, or it may be closely sort of wrapped around the stalk. And again, that's another critical feature. Cespitose, kind of a fancy word, but it's a useful one. Now, botany has a lot of technical terms that I think are really silly because there's perfectly good English words that mean the same thing and you don't need the Latin. So for example, if you get a book that's too technical, it'll use something like infundibuliform, which means funnel-shaped. So why not just say funnel-shaped? But it's not easy in a few words to describe cespitose. It simply means that the mushrooms are not only in a cluster, but the stalks all come from a common point. So you not only have a lot of them together, but if you trace the stalks back, they all meet Okay, well that's a lot of words to say that, so this one word, cespitose, simply means that. Okay, emerging from a common point. One term that can be confusing, it'll say that this species has narrow gills. That doesn't mean how thick they are. It means how deep they are from bottom to top. Okay, so broad or narrow refers to depth. Cuticle basically means skin, and so, and a really fancy word that some books use is pileopellis. That's the surface of the cap, so that skin-like surface that you can often peel off. Underneath that surface is what's called the context. It's between the skin and the gills or tubes. So if you cut an ordinary grocery store mushroom, underneath that skin, you'll see this clear white tissue, and then you'll see the brown gills. Okay, so that clear tissue there is the context. Hyphae, that's plural, hypha is the singular, those are these microscopic filaments of which all your fungal tissues are formed. Okay, so your whole mushroom, if you look at it microscopically, if you take a section, you'll just find a tangle of these microscopic filaments, and all parts of the mushroom are formed of them and organized in different ways. Collectively, when you consider all of the hyphae that gather nutrients for the fungus, that's what is called the mycelium, or mushroom growers would refer to it as the spawn for the mushroom. So that's the tissues from which the fruiting bodies eventually emerge when conditions are right. And it's those fruiting bodies that we call mushrooms. Then there's two kinds of veils, and these are also very important. Some mushrooms, not all mushrooms, have these protective veils that protect the spores when they're developing. One type of membrane just covers the bottom of the cap, and so it covers the gills or pores of the mushroom, and it basically is protecting the immature spores. Well, as the mushroom expands, eventually it grows horizontally, stretches that membrane so much that it ruptures. And that can leave this skirt type thing on the stalk. It can also, in some cases, leave these fragments around the edge of the cap. So that's referred to as a partial veil. A universal veil is a membrane that covers the entire mushroom, not just the base of the cap, but the entire mushroom, and it ruptures when the fruiting body grows vertically and stretches it to the point that it can no longer take it, and it ruptures. And that will, in some cases, leave this cottage cheese-like warts on the top, or in other cases, leave this cup-shaped vulva at the bottom. Okay? Now again, not all mushrooms have these. Some have one and not the other. But they're very important in distinguishing genera, and in particularly the genus Amanita, which is the really deadly one. So that's just a little bit of terminology. And I know vocabulary is boring stuff, but you really have to know at least that much in order to be able to read the field guides. Questions about any of those before we get a little bit more here? Okay, how are the gills attached? If you look at the very first page on your handout, I've got some drawings. And I'm not a very good artist, but I think these are good enough for our purposes. So at the top, you see a typical parasol-shaped mushroom. And so I've labeled the parts with these words that we just talked about. Now, this is an example of one that has both an annulus and a vulva. The genus Amanita, which contains the really deadly mushrooms that are most responsible for fatal mushroom poisonings, are in that group. So a typical Amanita looks like that. It's got a cup, it's got a skirt, it may or may not have warts on the top, but it has a very distinctive appearance. Now, let's talk about gill attachment. If you were to section the mushroom right down the middle, through the stalk and the cap, you would see various things. If the gills do not touch the stalk, you'll see this first drawing, the free gills. So if you imagine that now, this is just a section, but if you imagine the mushroom itself, that's going to be a clear space around the stalk. Okay. If you look at the grocery store mushroom, does it have such a clear space, or do the gills reach the stalk? It's one of those questions that when you're cooking you never think about. Well, it turns out they do have free gills. Genus Agaricus does have free gills, so there is a clear space. And next time you take a grocery store mushroom, look at it. It's very obvious. Okay, now, if it's not free, then that means the gills must come over and reach the stalk. How do they do that? Well, if they come in straight, so more or less perpendicular to the stalk, they're called adnate. Okay? Comes in perpendicular to the stalk. If they go up a little bit and then come back down, they have a notch in them before they hit the stalk, they're called sinuate or notched. Okay? So they go up toward the cap, but then they come back down just before they hit the stalk. If the angle between the stalk and the edge of the gill is less than a right angle, so if this is the stalk, if the gill comes in like that rather than like that, it's called, in some texts at least, adnext. It's a nasty term because it's so similar to adnate, it's hard to keep them straight. And moreover, some books are not really quite consistent on it, but most books, adnext means comes in at that kind of an angle. So think of three possibilities. If it comes in at a right angle, it's adnate. If it comes in at less than a right angle, it's adnext. If it comes in at more than a right angle, that means it goes down the stalk, it's decurrent. Okay? So on your little chart here, I've tried to indicate what a notched gill looks like. I could probably do a little better job on that. And decurrent gills means that it's definitely coming down with more than a right angle there. Any question then about those kind of technical terms? It takes a while to learn them, but again, you will find them used constantly in the book, so you need to know them. Okay, well, let's talk a little bit about mushroom poisoning, because mushrooms, of course, have a real bad reputation. I want to point out that if you just go out and you collect plants at random and you cook them up and eat them, you are just as likely to get poisoned as if you do that with mushrooms. In fact, you may be more likely to be poisoned because there's a lot of poisonous plants out there, and actually the number of seriously poisonous mushrooms is quite small. If you do that with a plant and you don't get one of the really deadly ones, then of course you'll have a bad experience, as same is true of course with a mushroom. But the difference is the latency period, the time between when you eat the plant and you start to feel symptoms, is usually short. It may be very short, a matter of a few minutes. There used to be some plants right out here behind this building. And I'm trying to think of the name of them. Somebody remind me, stuff that Socrates was... Hemlock, yes, water hemlock. There used to be some water hemlock growing out here behind. That's a really deadly plant. Well, if you eat some of that, you'll develop symptoms very quickly, and the problem is to get you to the emergency room before the stuff kills you. But the problem with the mushrooms, and I think the reason that they have the worst reputation, is that they're insidious. They have a long latency period. The worst of them, it's more than 24 hours. So you eat one, you have a good meal, you enjoy it, suddenly 24 hours later the symptoms develop. You may not even realize what caused them. And by that point, the stuff's out of your stomach. So pumping your stomach's not going to help. It's already in your system. That's why the deadly mushrooms are so deadly, because there's not too much you can do. Actually, there is an antidote that has recently been approved for testing in the country, and I'll talk a little more about that. Okay, but if you know what you're collecting, how risky is it that you're really going to get seriously poisoned? So let's look at the next step. This is taken from a book which is the current best reference on mushroom toxins. It actually has a section in the middle for doctors with detailed chemical information about what the toxins are, what the treatments are, and so on. And it has this chart this culinary classification of the fungal kingdom. So actually, I think that estimate at the top is probably on the small side, but on the conservative side. But more or less, he says 250,000 species of fungi, of which maybe 10,000 are these fleshy, large fruiting bodies that we call mushrooms, the others being microscopic things, like, for example, the athlete's foot fungus and things like that that don't form mushrooms. So there's maybe 10,000 fleshy macrofungi, of which there's probably 2,000 edible species and maybe 400 poisonous ones. Of those that are common, maybe 20 common poisonous species, and fewer than 6 common lethal ones. Okay, so it's not a large number. The other poisonous species being ones that would make you sick at your stomach or something like that, but not generally kill you. Then on the edibles, okay, maybe there's 100 species that are picked in any quantity, maybe 80 that are grown experimentally, and 40 of those, or half of the 80, that are actually cultivated, but only 5 or 6 that are really produced on any large scale commercially, and maybe 15 to 30 species that are commonly eaten. So I would say those of us in the mushroom club that have a lot of experience have probably sampled somewhere in the range of 15 to 30 species, many of which weren't all that impressive and we decided weren't worth the effort. Okay, so the number of really good edibles is pretty small. Okay, statistically, the majority of mushroom poisonings fall within 3 groups. Asian immigrants, especially Southeast Asia. Why is that? Because there is an edible mushroom sold in markets in Thailand and places like that that's almost a dead ringer for our destroying angel, the most poisonous of the Amanitas. They don't have our poisonous mushroom growing over there. But if they're immigrants and they come over here, they see it, they think it's the same thing they've eaten back there, they go out and collect it and eat it, and they're likely to end up a fatality. And usually when that happens, it's an entire family. It's usually mother, father, and a whole bunch of kids. So they're at risk because of this similarity. Children are probably the most common type of person that's taken to the hospital for having consumed a mushroom. The vast majority of the time what they've consumed is not something very harmful, but you can't take the chance. So children unfortunately graze on the lawn, whatever's there, and that includes mushrooms. They'll put almost anything in their mouth. So if you have a child and you happen to see this child eat a mushroom, you better take them to the emergency room just as a precaution and have their stomach pumped. Had the experience a few years ago of a couple call me and ask if they could come over with a piece of a mushroom because their child had eaten one and they'd already taken the boy to the hospital and had his stomach pumped, but they wanted to know what it was he'd eaten. Well, it turned out he'd eaten a honey mushroom, which is one of the ones that a lot of us go out and enthusiastically collect, so this kid had good taste in what he ate. Now, it's really unfortunate he had to have his stomach pumped, but I reassured him, you did the right thing. I mean, if you don't know what it is, you can't afford to take a chance, so you did the right thing. The third group, for whom I have no sympathy at all, of those who are likely to be poisoned, are those that are seeking a psychedelic experience. They're looking for a hallucinogenic mushroom, and they're not very good at identifying things, and they eat one of these little brown things, and it happens to be a conocope, which is deadly poisonous, and now they're on life support and on the liver transplant list. Okay. So, there are precautions that you should observe. Obviously, don't even think about eating it unless you've identified it to species. If you have any serious question about what species it is, don't take a chance. And especially, make sure you've taken a spore print, because again, some of these look-alikes you can distinguish that way. You should try just a small amount the first time and save some uncooked. In the case that you made a mistake, you start to feel sick, and you need to have a specialist figure out what it is. A specialist would much rather have you show up with a piece of specimen that they can look at to identify it than have to examine spores in your vomit, which unfortunately some people end up having to do. So try just a small amount the first time. I can tell you from personal experience, there are some mushrooms that are listed in the books as edible with caution. And I once tried one of these. I had no doubt of what species it was, but edible with caution means that some people are sensitive to it. So I observed this precaution. I had a small amount, and no problem. It tasted good. I didn't have any problem. The next day, I had a larger amount, and about 2 or 3 hours afterwards, I started feeling nauseous, and eventually threw up. Well, I knew that the dangerous ones have a long latency period. So although it wasn't a pleasant experience, I was not worried. I knew I happened to be one of the people that's sensitive to this particular mushroom, so I won't try eating it again. But this can happen. If you observe this precaution, at worst, okay, you may have a bout of nausea. But then it'll be over with and everything's okay again. Anyway, you save a little piece in case things do turn out worse. And hopefully someone will not eat the mushroom. They can serve an observer and be able to get help if it turns out to be a serious poisoning. Again, it's not very likely any of this will happen, but you might as well be safe. Okay, don't eat mushrooms raw. Morels are toxic if eaten raw. So don't eat them raw, okay? You can get away with it with button mushrooms, the commercial button mushroom, but that's one of the few that you can really do that. If you haven't tried a mushroom before, you shouldn't consume it with alcohol. Many mushrooms are okay, but there's one particular genus called Coprinus, which is common, and one species in particular produces a chemical called antabuse, which is actually a drug that they use in alcoholism treatment programs to get people to quit drinking. And what it does, if you consume alcohol and you've got this drug in your system, your whole body turns lobster red, you start sweating profusely, you feel heart palpitations. It's a very frightening experience. The antidote turns out to be reassurance, because it does eventually pass. But apparently it's quite frightening, and so there are some mushrooms that produce this chemical antabuse, and if you happen to eat one of those with alcohol, you could end up being very, very concerned about what's happening to you. And of course, you shouldn't sample, I should say, more than one species at the same time, because then if you have a problem, you don't know which species is responsible for it. Of course, make sure you don't consume any ones that are started to spoil. Use your usual precautions about cleaning and cooking. And you also have to be careful to avoid contaminated collection sites. Used to be in the days of leaded gasoline, they warned you about not collecting mushrooms next to a busy highway, because there were actually people that developed lead poisoning from deposition of lead from the fuel. That's not so much a problem anymore, but there's a guy that I know well in the New Jersey Club who is currently on a list to receive a kidney because he's suffering kidney failure, not from the mushroom per se, but he collected morels for years from old apple orchards, not aware that some of these orchards had been sprayed with pesticides that contained arsenic. And arsenic's a cumulative poison. It doesn't show up until it reaches a certain level. Well, eventually he developed heavy metal poisoning, and it took him a while to identify it, but they finally figured out he was suffering from arsenic poisoning. And so he had to go through a period of chelation therapy, which got rid of the arsenic, but unfortunately the chelation therapy damaged his kidney. So he's now on a kidney transplant list. So you do have to be careful sometimes to know a little bit about the sites. Apple orchards are one of the places they claim that morels are found, and they pretty much have to be old apple orchards because the more modern ones, they spray them with fungicides, so you won't find mushrooms there anyway. But even with the older ones, there is this danger, and they have actually, after this happened, they went back and sampled, the next summer, they sampled various of these places that he'd been collecting morels over the years, and they did indeed find a positive correlation with the arsenic in many of those sites. So you have to be careful. Even with mushrooms you buy commercially, you can run into a problem. Our club almost had a problem a few years ago when we bought some truffles from Oregon that for some reason or other turned out to be contaminated with some staphylococcus. And because we put some of them in a soup that was just barely cooked, 4 of our officers, including me, ended up with some nasty nausea from the staphylococcus. It had nothing to do with the mushrooms, but they hadn't been properly processed.
It came on right away.
Yes, it came on very suddenly. I mean within 15 minutes, which told us right away this is not a mushroom toxin. This is something else, more likely a bacterial or... Okay, well, you've listened to me talk a lot. Let me show you some pictures now of some, starting with a particularly dangerous species. This is Amanita phalloides. It is really a European species, and up until about 20 years ago, it didn't exist in the U.S. Somehow, spores made their way over here, and it was first discovered, I think, down in the Carolinas, and it has gradually moved northward. It's not common. The first time I ever saw it, though, was in a lawn about 3 doors down from where I live. And I was actually excited. I knew what it was when I saw it, and I was excited to find it because I knew it wasn't common. Last summer, we found some of these in Codorus Park. So they're a tall mushroom. They have a greenish cap. They have what to me is a very unpleasant odor. I can't understand why anybody would be tempted to eat one of these things. They have 2 important characters you can see right here. Notice the skirt, and it's not just a ring. It really hangs down like a woman's skirt. And they also have a volva. Notice that cup at the base on both of those specimens. Okay? It turns out if you take a spore print, it's a white spore print, and they also have free gills. If you see a mushroom that has those 4 characters, it's got a cup, a skirt, white spore print, and free gills, leave it strictly alone because it's going to be one of these deadly amanitas. Now, not all amanitas have the skirt and cup. Not all of them have free gills. All of them do have white spore prints. And there are some amanitas that are edible, and even some that are considered delicious. But I recommend you don't eat any amanitas because it's a large genus, they're hard to tell apart, and the risks are just too high. Okay, that one is less likely that you'd encounter than this one. This is Amanita bisporidura. It's called the destroying angel. It's a beautiful mushroom. It grows primarily in woodlands, leaf litter. It's a ghostly white. It's a large, impressive looking mushroom. Again, it has a prominent cup at the base. It has that skirt that hangs down. It has free gills and a white spore print. So those 2 have the same toxins in them, and this is the one I was talking about that is toxic to the level of 0.5 milligram per kilogram of body weight. Okay, so it's very, very deadly. This is the one that shuts down your RNA polymerase and will start showing symptoms after about 24 hours and put you on the liver transplant list.
It doesn't look like it has a pretty gill. Can you only tell when you cut a section through it?
No, actually, if you look really closely, I think maybe you can see it on this specimen. I agree, it needs to be in a little sharper focus. But there is, in fact, if you really look at them in the field, it is obvious that the gills go up into the cap and leave a little space. So it's the photo really that's the problem here.
It's like a radius.
Yeah, it actually curves into the cap, and when you look at them, you hold it with the stalk towards you, you'll see this free space around it. Okay, so again, the characteristics of those 2 deadly Amanita species, the white gills, which are free or nearly free, white spores, technically, if you really want to confirm things, and if you have to go to a hospital, the spores turn blue in what's called Meltzer's reagent. That is something that mycologists use. It's a mixture of potassium iodide, iodine, and chloral hydrate. It's very useful. Unfortunately, since 9/11, it's extremely difficult to get because it's a controlled substance. Chloral hydrate is knockout drops. It's a date rape drug. So you can't get Meltzer's reagent. Even though Meltzer's reagent itself is not a controlled substance, chloral hydrate is, and so it's hard to get, but it is a reagent that's almost essential in some mushroom studies. And within the amanitas in particular, some have spores that turn blue, they're called amyloid. Some have spores that turn red, and some have spores that don't change color at all. So that's a technical chemical point. The cap is smooth. It doesn't have any striations, that is grooves that go out radially. And there's no veil remnants that hang off the margin. The skirt is pendulous. It's up toward the upper part of the stalk. And this cup at the base is firmly attached and free from the stalk at the top. This is why it's so vitally important to get the whole mushroom, because if you just somehow picked it up, picked it off, and were careless, you might miss getting the cup. You might just simply take it off too short and miss the fact that it had a cup there, and you've eliminated one of these critical identification features. Okay, here's another mushroom that contains the same toxin as those two amanitas. Now this is actually a fairly small mushroom. It's no more than an inch in diameter. So this is an extreme close-up here. It's also usually a little darker brown. It's awfully hard to photograph mushrooms because they like dark places and they like to grow low to the ground. This usually grows on decaying wood, especially wet wood. Its scientific name, Galerina autumnalis, suggests that it comes up in the autumn, which it does, but it also comes up in the spring. So if you find it in the spring, you don't just assume that's not what it is. This is one of those little brown mushrooms that I've been warning about. It does have a ring on the stalk, but notice it's not a pendant skirt. It's just a ring. So it's a small brown mushroom with a ring on the stalk. It's brown spored, grows mainly on wet woods in the spring or fall. Because it's rather inconspicuous, it's not very likely to be collected. And I think that's the reason that very few people end up in the hospital because of this, because not many people would eat it. But it does have a look-alike, which is called the winter mushroom, which does not have the ring, and which has white spores instead of brown spores. So if you take a spore print and if you observe the ring, you're not going to make that mistake. But if you're careless and you pick what you think is the winter mushroom and you get one of these, it's just as deadly as the amanitas. Okay. So again, its characteristics: it's small brown one, grows on decaying wood, has a ring which may, however, get rubbed off or washed off, gives a brown spore print, resembles this edible species, Flammulina velutipes, but that species that's edible has no ring, gives a white spore print, and has this. The deadly one has the same toxins as the amanitas. Okay. Now, this is a very common mushroom. It's another amanita. It has many of the characteristics we've seen before. It has a white spore print. This shows you some immature ones and ones that are starting to open up. It doesn't exactly have a cup at the base, but notice the sort of rings or ridges, I should say, at the base. So it is considered a vulva, but it doesn't look like it did on the others. And this one also has those cottage cheese-like warts. So in this case, what happened to that universal veil, instead of most of it staying on the bottom and forming that elaborate cup, most of it got broken up in pieces on the top. And so the cottage cheese stuff is the remnants of it. This one is not deadly poisonous. It's actually hallucinogenic, but it's also a really nasty gastric toxin. So you get some pretty awful nausea with it, and even the potheads that are going out there looking for trips don't tend to eat this because it's just too unpleasant an experience. It's spectacular, though. Now, this shot was actually taken over in Pine Grove Furnace, and it's kind of unusual around here because this one's red, and in the Rocky Mountains, it's absolutely blood red in color. Most of them here are orange or yellow rather than red, so this one was kind of unusual. But anyway, it's called the fly mushroom. Musca is Latin for fly, and the reason it's called that is apparently the claim is the ancients used to make a decoction of this as a fly poison.
Yep.
So that's what it comes from.
Telling me.
Yep.
Back home, we used to sprinkle sugar on them.
Okay.
And
The flies were around all day, you know.
Okay. Yeah. Yeah, so Muscaria in Latin is fly, so the fly mushroom or the fly agaric. Okay, now this one I want to concentrate on because this one's very common. As a matter of fact, just last week I saw a large fruiting of this in a neighbor's lawn. It has a complicated Latin name, Chlorophyllum molybdites. This is the one that has a green spore print, extremely unusual. It's a light lime green, or almost it's like pistachio ice cream. It's that shade of green. However, when it first opens up, the gills look completely white because the spores haven't yet matured. The gills will turn a dark green eventually, but at first they look white. Now this is before it opens up. Notice the scales, the really prominent scales. These aren't warts. They're not like the cottage cheese stuff we saw on the previous slide. These are scales. They look like you could just peel them off. Okay, now that will open up. So this is unexpanded. If we go to the next slide, you'll see what this looks like when it opens up. It's huge. These things are almost dinner plate size. They're really large. They grow in grass, in urban lawns primarily. They have a little bit of a knob in the center. The technical term is an umbo, U-M-B-O. And they're darker in the center. They're brown because there's kind of a concentration of the scales there. And if we look at the next slide, we'll see the underside, what it looks like. It has a very prominent ring, which is actually sort of fringed and double-edged. It's got an upper as well as a lower portion to it. It's also movable on the stalk. You can move the ring up and down. It's not actually attached. And this one has finally started to show the greenish tinge to the gills. This mushroom is responsible for more hospitalizations than any other. Even though it's not deadly, it produces such cholera-like diarrhea, I mean just incredibly watery diarrhea, that you have to be put on IVs to restore your fluid balance. It's a very, very unpleasant experience, the terrible nausea and diarrhea. So it's responsible for more. Now it is a lookalike for one very good edible mushroom. However, first of all, that edible mushroom has white spores, not green spores. It looks very, very similar, except the ring is a little different shape. But fortunately, this one that it's a lookalike for is actually quite rare. I've only seen it about twice in 20 years. So if you see one of these, and especially growing in your lawn, you can pretty much bet that that's what it is. And if you take a spore print and get that green spore print, you know for sure that it's this chlorophyllum. So it's a very dangerous mushroom. And if I had a child, that's the one I'd worry about. The others, the Ammonitas, are primarily woodland mushrooms. But if these big things come up in your lawn and you have a child, get rid of these things. Mow them down or do something.
The picture you showed of the spore print, it looked like you had cut it off the stem so you can get the full circle.
You can do it either way. A lot of people just cut off the stem and lay the cap down. If you don't want to do that, if you want to preserve some of these features, make a little slit in the piece of paper, slide it around the stem, make a little hole and slit, and then you can keep the thing whole and still get your spore print. And what I'd do, I'd just suspend it in a water glass. So put the paper on the rim of the glass, cap above, and the stalk down through. Okay, so characteristics. Again, it's common in lawns, it's very large, has conspicuous brown scales, this annulus that does not wear off, it's persistent, it's double-edged, has this lime green spore print that's extremely unusual, does closely resemble this edible species, Macrolepiota prosera, but the latter's rare and has white spores. So you can look in a book, you can see what Macrolepiota prosera looks like, and you'll see that it is very similar, but it's not one you have to worry about because it is so uncommon. Now, this is another one. It's one of the few that has a common name. It's called the jack-o'-lantern mushroom. Not so much because of the color, which is sort of pumpkin colored, but it's bioluminescent. It glows in the dark. This is fairly common in late summer. And amazingly, and you'll see why I say that in a moment, this has been mistaken for a chanterelle. Now later I'll show you what a chanterelle looks like. Keep this one in mind when we look at it, and then you tell me if you think it really resembles a chanterelle in anything except color. This is a gilled mushroom. It grows usually at the base of trees, and this is a good example of one that's cespitose. That is, the stalks all come from this common point. Chanterelles are not cespitose. They may grow in groups, but they each have their own separate stalk, and they do not come together, okay? And chanterelles are funnel-shaped, and they don't have gills, they have ridges. This violates all of those. This thing has gills, it's cespitose. I don't know how people could make that mistake, but this is another one that'll give you a nasty nausea bout if you eat it. So, as I said, that some people have made the mistake. I don't know why, other than the color. If you can't distinguish something like this from a chanterelle, you really shouldn't eat wild mushrooms at all. You should leave it to other people because you're obviously not discriminating enough. Okay, another one that can be dangerous, we saw this name before, this hard skin, the yellow hard skin. Some people call this the poison pig skin because the surface kind of looks like a football. It's a small mushroom. It's no more than 2 inches across usually. Looks sort of like a puffball. Technically it's called an earth ball rather than a puffball because it doesn't puff. That is, it doesn't have a pore through which the spores are ejected. Rather, eventually that hard skin will rupture in a sort of a crack and expose the spores. But the skin is much harder than it is on a puffball. And although the interior is white to begin with, when you cut it with a pocket knife, if it's mature, it'll be almost purple, black or purple. True puffballs, the interior is a uniform white, like a marshmallow. It may eventually, as they age, turn to sort of a tan or brown, at which point they're not edible anyway because the taste is bitter. But unless it's really young, you wouldn't mistake this for a puffball if you cut it. And that's one of the things I always warn you with puffballs. Even though puffballs are safe, you might, if you're not discriminating enough, get a button stage of something else and think it's a puffball. But if you cut it in half, you can tell whether it's a puffball or, for example, a button stage of an amanita, because if it's an ordinary gilled mushroom and you cut it, even when it's in the button stage, you will see the shape of cap, gills, and stalk when you cut it. You will actually see those structures inside when you section it. In a puffball, it'll just look like a uniform marshmallow. So this is an earth ball, and again, this one is a nasty gastric experience. Creates what's called gastric awareness, acute gastric awareness. Okay. All right, now this is a false morel. Okay, this is the one that produces a toxin that turns out to be a rocket fuel oxidizer. So this is the one that they learned about through the space program. This is a dangerous mushroom because it's been sold on markets in Michigan, for example. And about 99% of the time, it's edible. The other 1% of the time, it's fatal. What's going on? Well, it turns out individual specimens vary a lot in the concentrations of this toxin. Secondly, it's a cumulative poison, like arsenic, so you can eat it many times and not have a problem because you haven't reached your threshold. But it also has a very sharp threshold, so that once you reach that threshold, if you go beyond it just a little bit, you're suddenly dead. So it's very dangerous. It should never be eaten or sold, but people get lulled into a false sense of security because they've eaten it before and they haven't had a problem. Those of you that know what morels look like, do you think this resembles a true morel? A true morel is pitted. This is not pitted. This is brain-like. It's convoluted. It's wrinkled. Very, very different. We'll look at a shot of a true morel later, but they come up at the same time that morels do, and I think that's why maybe they're called false morels. Around here, they're actually quite a bit less common than the true morels. I've not seen very many of these, but anyway, that's a false morel, and they are dangerous. Well, let's look then, and those are the dangerous ones that I think you need to learn first. Then look at some of the common safe ones. Okay, these are oyster mushrooms. so-called from their shape. They're a gilled mushroom. They have white gills. It's actually a complex. It's not just one species. It's several closely related species, but they're all edible. They do have a short stalk, but it's off-center. So these grow on the sides of trees. They have a very short stalk that sort of comes out, not to the center, just attaches the mushroom to the tree. They generally grow in layers, one above the other, often quite a lot of them. It's an extremely good mushroom. The only one that you might mistake it for, there's a very sort of fragile mushroom that's white. It's much, much thinner than this. It does have gills, and it's white and sort of shell-shaped and grows on the side of trees, called angel's wings. I don't have a picture of it here. Recently, there's been some discoveries that apparently some of them can be toxic, although I don't think there's any U.S. Cases. But it's actually pretty easy to tell apart. And so oyster mushrooms are plentiful. In fact, they're even growing them commercially now. You can buy them in the grocery store. And they're a very good edible. I don't think they taste like oysters, but they look like oysters in the shape. These are true chanterelles. There's two kinds here. This is not the best photo because this has been transferred from a slide to a digital, but those are yellow chanterelles on the left and so-called black chanterelles or black trumpets on the right. You can see, especially in the upper ones here, you can see that they have these ridges, but they're not gills. Okay? And these things grow singly, so they don't grow cespitose. They are that same orange color we saw in the jack-o'-lantern mushroom, but I don't think there's any resemblance. These are funnel-shaped, and especially the black ones are funnel-shaped. So they have these little ridges or wrinkling on the underside, but not true gills. They, in good year, are plentiful in the summer, especially the yellows. They are excellent edibles, and I think they're pretty unmistakable. But again, if you think these look like that jack-o'-lantern mushroom, then I wouldn't go out and collect chanterelles because you're not discriminating enough. This one is an unusual one. Most species in the genus Antiloma are poisonous. This one is not. And the way you can tell that you've got it, because this is a tough genus to work with, are these misshapen things that grow along with it. Okay, so you see these odd things over there? Well, it turns out that these mushrooms are aborting the honey mushroom. That is, they are actually parasitizing another mushroom, the honey mushroom, which is one of the good edibles. And the result of that parasitization is that strange lumpy thing. But only this species does that. So if you see those, then you know it's this Antiloma, and it is a safe edible. And in fact, even though they look strange, if you cut those odd-shaped lumps, if you section them, you'll see little nodules, little sort of pink nodules that look a lot like pearl tapioca. And both they and the regular Antilomas are edible. And a lot of people around here collect them avidly. So it's a strange one, but because it is so odd, it's distinctive.
How do they abort the honey mushroom?
The mycelium goes down and attacks the mycelium of the neighboring honey mushrooms.
Whether it's fruited or not.
Yeah, yeah, and then it causes the fruit to be aborted, but it's really attacking the mycelium of the mushroom. Okay, there's a number, again, a complex called chicken of the woods. They're closely related. There's maybe 6 species. They are a polypore. So this species tends to grow on stumps. This was at the top of a stump. Generally, you find them at the bases of trees. They're either orange or yellow, and they have this lighter rim. It's mostly the rim that's edible because the other part tends to be very tough, and it does require long cooking. Many people think they taste like chicken, but then that's a sort of a common phrase to say something tastes like chicken, whether it really does or not. It is a good edible, the part that's not so tough. The only thing you have to be careful of is whether it's growing on a hardwood or a conifer. Most of the ones around here are growing on hardwoods, and they're okay. In California, there's some that grow on conifers, and although the mushroom isn't poisonous, it absorbs something from the conifers, I don't know, from the resin or what, that makes people sick at their stomach. So I don't know whether it's pine sap that they're ingesting or what, but at any rate, if it grows on a conifer, you should leave it alone, not because of the mushroom itself, but because of what it's absorbed. These can be spectacular. I saw one over in the park in Wrightsville last summer that was absolutely magnificent. It was just a huge cluster, and apparently nobody had seen this. Nobody had damaged it. I got a lovely photograph of the thing. I was sitting down just eating my lunch on a picnic table, and I just looked over and saw this orange thing some distance off, and I thought, you know, that's got to be something interesting, and it was an enormous one of these. Okay, then there's also hen of the woods, not to be confused with chicken of the woods. It's called hen of the woods because it looks like a hen sitting on the nest of eggs with the feathers ruffled out. It grows at the base of trees. It's quite common around here. It's large, and by large I mean, you know, you can get some this big around. And although it's not real easy to clean, it has these layers, and they're very good. So at our tastings, we often have dishes made of this Griffula frondosa, which is the hen of the woods. Okay, here's a true morel. Now again, does that look anything like the false morel that we saw before? I don't think so. It's pitted. There are several species. This is the early one that tends to come up first, the so-called black morel. Here's another little bit of Latin. CEPS is another ending that means cap, or head, actually. And angusta means narrow. So it's the narrow-headed morel. Okay. And then the next picture, one year I and a couple of other club members went down to Great Smokies, and we collected some 400 morels. And this is just a mixed collection. This is what we got one day. And so you can see the variety here. Some of these are light colored, some of them are dark. The shape of the head varies from elongated to not so much. So there's several species of morels that are common, but they're all equally good and equally delicious. So they're one of the safest of your mushrooms, as long as you cook them. Again, don't eat them raw. And then the giant puffball. Again, there's many species. There's some that are pure white and almost basketball size. I found a couple of those last year. This one is the more common one. This one is usually softball size, and it's not white. It's got this mottled pattern on the surface. But if it's fresh, you cut it, and you'll see that it's a pure white marshmallow-like thing. And so you slice it and you fix it sort of like French toast or potato cakes. And it resembles tofu in texture and somewhat in flavor, although a little stronger flavored than tofu. So it's very mild flavored. And then these are some of the smaller puffballs. Again, they're edible. It takes an awful lot of them, though, to make a meal, and I don't think too many of our members actually collect these very, very actively. Again, they're mild flavored, and they're kind of more trouble in a way than they're worth. But they are safe.
And that little round thing at the top.
The umbo, yeah. That's where the pore will develop. And so these are pretty young ones, pretty fresh, but in fact, the one I think you can actually see a pore. The one right here. See that little pip? Mm-hmm. Right in the center, I think.
The one above it.
And yes, that one too. I don't think that's one of the little spines. I think that's actually the pore that's developing. Some of these species do have these little minute spines on them, and there's quite a few species of puffballs, and they're not that easy to tell one from another, but fortunately, they're all equally edible, so it's not about golf ball size. Yeah. Now, I mentioned that there were these tooth fungi. This one is also, the ones I've seen are mostly about softball size. They grow on the sides of trees, and they're just covered with what at first looks like hair, but when you look at it closely, it turns out to be these icicle-like teeth. These are now sold in stores under the name hedgehog mushrooms. So they do, some of these specialty places do grow them commercially. You don't find these too commonly, but when you do, they're very good. So that's one to kind of keep a lookout for. Okay, let me talk a little bit about field guides now to kind of wind things up here. I mentioned the most comprehensive one is this Mushrooms Demystified. So what are the pros and cons on this one? Well, it's excellent in terms of its keys. It's got several different types of keys. It contains an amazing amount of information, and the guy is a very amusing writer. It has a good glossary of these technical terms and also these Latin combining forms, but it's too big to carry around in the field. It's a good reference on your shelf. Its author lives out in California, so there is somewhat of a West Coast emphasis. Some of the mushrooms in here you won't find here, but on the other hand, most of the ones we do find are in here. And the other disadvantage is it doesn't have very many color photos. It has a fair number of black and white photos, but it doesn't have very many color photos. So it's a reference to have on the shelf. If you really sit down and read this thing, and I must admit there's still sections of it I should read, by the time you finish, you will learn an awful lot about mushrooms, and you'll be pretty expert yourself if you make it all the way through this thing. So again, that one you can get from Amazon. It's somewhere here. I had a price on that. None of these are terribly expensive. They're all in around the $20, $25 range. Somewhere I have a list of the price. Anyway, it's one you pretty much would have to get from a place like Amazon, though, because it's a very small press, and if you try to order directly, it just takes forever, and most bookstores won't have it. So the easiest way to get it is online through one of the big outfits like Amazon. Okay, so let's go to the next page now. Now, the one that most of us carry in the field with us is this one, the Audubon Society Guide. So it's compact, you can carry it along with you. It has a nice waterproof cover. It has a large number of very good photographs, and it also has in the back half detailed textual descriptions. But what it doesn't have, or at least not much, is keys. It does have a little bit of a pictorial key in the front, but it's really not enough. However, online you can find an actual textual key, a dichotomous key that was made up later. So if you want to print that thing out from the online site, you can get a key. So anyway, there's the pros. The disadvantage is that it doesn't have much in the way of keys. It's also not arranged by families, and it has all these common names that are just made up. So when you're just searching by photos, it can be useful. But it's not real useful if you've got a mushroom and you have certain characters, you can't key it out. You don't know really where to look, except just in terms of the gross shapes. But it is a useful guide. Okay, Mushrooms of Northeast North America is this one. It's another small one with a waterproof cover. It's quite good. It does have very good keys and has good color illustrations. It does have a number of species that you won't find in the others. The main disadvantages are it doesn't have quite as many species overall, certainly not as the Audubon. And I found this thing to be poorly bound. It's starting to come apart in the binding. Now, of course, you can have it bound to avoid that problem, but it seems to be a little bit cheaply made. And finally, there's this basic reference, which is not a field guide. I highly recommend this. I've put thumb tabs on my copy, but it's called How to Identify Mushrooms to Genus, Volume 1, Macroscopic Features. So features that you don't need a microscope to distinguish. It consists of a lot of vocabulary. It has all these various technical terms. It has elaborate line drawings illustrating all these different ways that the gills can be attached. It's just an excellent source to get you acquainted with terminology and these basic stature types. It does have keys. It has tabular keys in the back. I think it's an extremely good book to give you background information, but it's not a field guide. It's not really intended, I think, as a field guide. But it does give you this detailed discussion of identification features, excellent drawings, and unlike most guides, its primary key structure is in terms of stature types rather than spore colors. So if you can't get a spore print, you can start with this one and at least get it down to a stature type, and you'll find one of these, you'll find it broken down by spore color after that, but at least you've got one row now, and you only have to look in that row for possibilities. So if you don't know what the spore color is, you've still narrowed things down. You're not stuck totally because you didn't get a spore print. This one, they actually have some leftover copies here that they're willing to sell. And I checked the price. Here, they sell this for $15 at the front counter. If you order it on Amazon, it's $19 plus postage. So if you're interested in a copy, you can get it more cheaply here than you can online. I think that's the last slide, isn't it? Check and see. Yeah. So basically, we're actually about 10 minutes over time. Are there any further questions that people have? Yeah.
Do you have any comment on whether harvesting the fruiting bodies of the mushrooms is neutral or negative to their population?
Well, if you just harvest the fruiting body in any quantity that a normal person would, I don't think it is. The real problem for conservation on fungi is habitat destruction. What happens is, people build houses or things like that on the growth places. Now, if you don't just take the fruiting body, if you actually dig up some of the mycelium, that's another matter. That is destructive, I think.
The spores from that fruiting body won't really impact the population.
Right. The probability that a given spore will germinate is pretty small. They produce them in the trillions. And actually, when you pick the mushroom, you will probably distribute some of those spores inadvertently anyway. In fact, when people that don't like mushrooms kick them over, they're actually distributing the spores. They're helping that mushroom come back. So, yeah, I don't think ordinary collection does. Now, if you're talking about the Pacific Northwest, where you literally have hordes of people go out armed to defend their territory, and they're collecting commercially, that's another matter. I think it is a serious issue there. Fortunately, we don't have that here. And you do not need a permit in Pennsylvania in any state park, state forest, or state game lands to collect mushrooms, nuts, or berries for your own personal use. And that's very unusual. Every surrounding state that I know of has restrictions on that.
We do have restrictions at the county parks, though.
Yeah, you do have restrictions here at the county park, but at the state level, you don't. Yeah. In Delaware, for example, you can't even get a permit for scientific collection purposes, which I find pretty amazing. But yeah, Pennsylvania is open on that.